Good morning, students. We will continue the carbohydrate metabolism. Today's competency is competency number 3.4 and 3.5 and topic is galactose metabolism. What is galactose? Galactose is a monosaccharide which is a aldohexose sugar and it is the fourth epimer of glucose. This galactose is present in milk in the form of lactose, and this galactose is absorbed from the small intestine with the help of sodium dependent glucose transporter 5 or GLUT5. In our body, galactose is exclusively metabolized in the liver. We will see this synthesis of galactose. Galactose is synthesized from glucose. First, glucose is phosphorylated to glucose 6-phosphate with the help of enzyme hexokinase, and here phosphate donor is ATP. Then glucose 6-phosphate is mutated to glucose 1-phosphate with the help of enzyme mutase. Now this glucose 1-phosphate combines with uridine triphosphate or UTP, which is a nucleotide. This UTP undergoes hydrolysis by removal of phosphate bond to form UDP, and this UDP combines with glucose to form UDP glucose, and two phosphate groups, one from glucose and one from UTP, are released in the form of pyrophosphate. This step requires enzyme UDP glucose phosphorylase. Now this UDP glucose is also called as active form of glucose, which undergoes epimerization to form UDP galactose. The enzyme is epimerase. Now then this UDP galactose is used for synthesis of lactose. So in this way, galactose is synthesized in our body from glucose. Now galactose present in the diet is utilized in our body by two different pathways. One is the main pathway of galactose utilization and another is alternate pathway of galactose utilization. First we will see the main pathway of galactose utilization. Now this pathway is present in the body since birth. Now in this pathway, the galactose is first converted into galactose 1-phosphate with the help of enzyme galactokinase. Here phosphate donor is ATP. Now this galactose 1-phosphate then combines with UDP glucose. Now these two compounds exchange their groups. UDP is transferred to galactose to form UDP galactose, and phosphate group from galactose is transferred to glucose to form glucose 1-phosphate. Here the enzyme required for this step is galactose 1-phosphate uridyl transferase. So this enzyme is very important enzyme of this pathway. It is also called as rate limiting enzyme of this pathway. Once this UDP galactose is formed, it may either converted into lactose or it may undergo epimerization to form UDP glucose with the help of enzyme epimerase. So in this way, the galactose taken in the diet is utilized in the liver with the help of this pathway. As I have already told you, galactose 1-phosphate uridyl transferase is the rate limiting enzyme of main pathway of galactose metabolism. Now second pathway of galactose utilization is also called as alternate pathway. Now in this pathway, the galactose which is present in our diet is first converted into galactose 1-phosphate with the help of enzyme galactokinase, and the ATP is the phosphate donor. So this step is similar to the main pathway. Now in the second step, the galactose 1-phosphate and UDP glucose exchange their group. UDP is transferred to galactose to form UDP galactose, and phosphate group from galactose is transferred to glucose to form glucose 1-phosphate. But here the enzyme required is different. The enzyme required for this step is galactose 1-phosphate pyrophosphorylase. In the main pathway, the enzyme required for this step is galactose 1-phosphate uridyl transferase. But in alternate pathway, the enzyme is galactose 1-phosphate pyrophosphorylase. Later on, UDP galactose is either converted into lactose or it is converted into UDP glucose with the help of enzyme epimerase according to need of the body. Now one point you have to remember about this enzyme galactose 1-phosphate pyrophosphorylase. This enzyme is active in the liver after 4 to 5 years of life. That means galactose is utilized through the alternate pathway after 4 to 5 years of life. Until that, the galactose is utilized in the children with the help of main pathway only. Now, significance of galactose metabolism. As I have already told you that galactose is exclusively metabolized in liver. So, galactose tolerance test is used to assess the functional capacity of the liver. Second, UDP galactose act as a galactose donor for various synthetic reaction. UDP galactose is a active galactose molecule which act as a galactose donor during various synthetic reactions. These reactions are synthesis of lactose, synthesis of glycose aminoglycans, means mucopolysaccharide synthesis, which we have studied during chemistry of carbohydrate. Galactose is also required for synthesis of glycolipids and cerebrosides, which we have studied during the chemistry of compound lipids. Galactose is also required for synthesis of glycoproteins. So in this way, galactose is used for synthesis of various compounds and the donor of galactose is UDP galactose. Now come to a very important disease related to galactose metabolism, is called as galactosemia. Semia means whenever there is increase in the galactose concentration in the blood, it is called as galactosemia. It is an inborn error of metabolism. This galactosemia is due to deficiency of two enzymes. Either it is due to deficiency of enzyme galactose 1-phosphate uridyl transferase, or galactosemia may be due to deficiency of enzyme galactokinase. So this is the two steps where these two enzymes are required. So either galactokinase deficiency or galactose 1-phosphate uridyl transferase deficiency leads to a disease called as galactosemia. First we will see galactosemia due to deficiency of enzyme galactose 1-phosphate uridyl transferase. Incidence given in the book is 1 in 35,000 live birth. Here, due to deficiency of enzyme galactose 1-phosphate uridyl transferase, galactose 1-phosphate is not utilized. See this figure. Due to deficiency of enzyme galactose 1-phosphate uridyl transferase, galactose 1-phosphate is not further utilized. Hence, galactose 1-phosphate will accumulate in the liver. When this galactose 1-phosphate accumulate in the liver, it inhibits the two enzymes in the liver. First is galactokinase, immediate enzyme of this pathway, and therefore galactose is not converted into galactose 1-phosphate, and hence the galactose level in the blood increases, leading to galactosemia. Secondly, accumulation of galactose 1-phosphate in the liver also inhibits the another important enzyme called as glycogen phosphorylase. Now this glycogen phosphorylase is the rate limiting enzyme of glycogenolysis pathway or glycogen breakdown pathway. As this enzyme is inhibited, glycogen is not broken down during the fasting state of the body, and hence such persons will suffers the episodes of hypoglycemia or it develops hypoglycemia. Now the clinical features of galactosemia includes First galactosemia, that is accumulation of galactose in the blood. Then galactosuria, when the concentration of free galactose in the blood increases, it is filtered into the glomerulus and excreted into the urine, leading to galactosuria. Also, due to accumulation of galactose 1-phosphate in the brain get damaged, leading to severe mental retardation. So severe mental retardation is due to accumulation of galactose 1-phosphate in the brain. Next clinical feature is hepatomegaly, that is enlargement of liver. Again, galactose 1-phosphate get accumulated in the liver and damage the liver. Jaundice is the next clinical feature, and it is mainly due to damage to the liver. When the liver is damaged, bilirubin uptake and its conjugation process is decreased. Therefore, unconjugated bilirubin level is increased in the blood, leading to jaundice. The most characteristic feature of galactosemia is congenital cataract. Cataract means opacity of the lens of eye, and congenital means present since birth. When the cataract is present since birth, it is the characteristic features of glycosemia. Why this cataract develops? This is because when the galactose level is increased in the blood, more and more galactose enters inside the lens, and when the more galactose enters inside the lens, this galactose absorbs water by the process of osmosis in the lens, and due to which this galactose and its reduced compound dulcitol get accumulated into the lens, leading to cataract. Next clinical feature is amino aciduria. When galactose 1-phosphate is accumulated into the renal tubules, it causes damage to the renal tubules, leading to excretion of amino acids from the tubules, a condition called as amino aciduria. So all of you seen that up till now whatever clinical manifestations we have seen in galactosemia are due to deposition of galactose 1-phosphate. Now diagnosis, as I already told you that congenital cataract is the characteristic feature of galactosemia. Apart from that, galactosuria is there, galactosemia is there. Prenatal diagnosis can be done by amniocentesis. It is useful. Now come to the treatment part of galactosemia. Only an effective treatment of galactosemia is withdrawal of lactose from the diet or withdrawal of milk from the child diet. Most of the symptoms after withdrawal of lactose get recede, but mental retardation once established will not get improved. Therefore, early detection of galactosemia is important. Lactose free diet may be continue up to 4 years. After 4 years, we have seen the alternate pathway of galactose utilization starts, and the child can utilize the galactose by alternate pathway. So galactose free diet should be continue up to the 4 years, and slowly after 4 years you can add the galactose to his diet. So this is the alternate pathway. The galactose 1-phosphate pyrophosphorylase enzyme get active after 4 to 5 years of life, and galactose is utilized by this alternate pathway. So after 4 to 5 years you can give the galactose to the child. Now come to second cause of galactosemia, that is galactokinase deficiency. It is seen in 1 in 40,000 live birth. Here the symptoms are mild. When galactokinase is deficient, then galactose is not converted into galactose 1-phosphate. Therefore, galactose 1-phosphate is not formed. Only galactose level is increased. As the galactose 1-phosphate is not accumulated in various tissues, the symptoms are mild, but cataract is seen due to deposition of galactose and its reduced form dulcitol into the lens. So this is about galactosemia. Now we will see a case which is frequently asked on this topic of galactosemia. One of these case which I have asked in the university exam is given here. The case is 1-year boy developed cataract, mental retardation, delayed milestones and hepatomegaly. Urine show Benedict test positive. They have asked you the question like, what is the probable diagnosis? Whenever you see 1-year boy developed cataract, that means it is a most probable case of galactosemia. Along with it, they have given mental retardation, delayed milestones and hepatomegaly. These symptoms also favors the diagnosis of galactosemia. They have asked you second question, name the deficient enzyme. Here galactosemia is due to deficiency of two enzymes, either galactose 1-phosphate uridyl transferase deficiency or galactokinase deficiency. But if galactokinase deficiency is there, then it does not cause mental retardation, hepatomegaly, etc. Therefore, the deficient enzyme in this case is galactose 1-phosphate uridyl transferase. Now, what is the cause of cataract? The cataract is due to accumulation of galactose and its reduced form dulcitol into the lens, which causes osmotic effect inside the lens and leading to opacity of the lens. Write the reaction catalyzed by the effective enzyme. In this case, you have to write down this reaction: conversion of galactose 1-phosphate to UDP galactose with the help of enzyme galactose 1-phosphate uridyl transferase, which require UDP glucose as a donor of UDP. So this reaction you have to write down in this question. What is the treatment for the disease? Just I have already told you that you have to withdraw the milk or lactose from the diet up to 4 to 5 years, because after 4 to 5 years the alternate pathway of galactose utilization becomes active and it will start utilizing the galactose. Therefore, the treatment is we have to continue lactose free diet up to 4 to 5 years. This is about galactose metabolism and its clinical significance. Thank you very much.